Good morning, everyone. It's cold, isn't it? Yeah. So first, I would like to thank the organizers for giving me this opportunity to talk about our technology. I'm from University of Louisville, Matoba. I'm talking about an EPICERTIN, which is a protein which has mucosal healing activity we engineered. So among many different potential applications, one indication that we currently focus is ulcerative colitis. It's a type of inflammatory bowel disease. It's a chronic disease, essentially, and with unknown etiology. But the long-standing ulcerative colitis is known to significantly increase the risk of developing colorectal cancer. So there are many cases worldwide with, this is a more industrialized country's problem with increasing rate of incidence rate, and particularly in North America, Northern Europe, with 5 million cases worldwide, with United States alone, 1 million patients, and with, incidence rate is increasing. Right. So you see drug market is huge. $7.5 billion worldwide in 2020, and with $5 billion of sales in the United States market alone. There's currently no cure for inflammatory bowel disease and ulcerative colitis. Conventional treatment strategies simply aim at blunting inflammation. However, the problem is that nearly half of patients become unresponsive, non-responsive to primary and secondary treatments. Therefore, there's a clear unmet need for developing new therapeutic agents, particularly that can go and beyond just blunting inflammation. So our product, EPICERTIN, it is a variant of oral cholera vaccine antigen. More specifically, it's a non-toxic component of viral cholera toxin B subunit. It's been genetically modified by our group to contain an endoplasmic reticulum mutation motif at the C terminus. So what it does is to make this protein exerting a very unique mucosal healing activity. We've shown in multiple publications that the EPICERTIN can induce mucosal healing in chronic and acute colitis mouse models, and it's been also confirmed by a CRO. And also we've tested its activity in human colon tissues isolated from IBD patients. And then also we've tested its efficacy in hamster model of radiation-induced oral mucositis performed by a CRO, all demonstrating that this EPICERTIN exerts a very unique mucosal healing activity, unlike the original cholera vaccine antigen. And also, we've shown that this protein can be very efficiently manufactured in conventional bacterial fermentation system. We also showed that this protein can be expressed in tobacco plants. It's a more innovative side of production, but we are looking to find a manufacturing partner using bacterial fermentation for the initial development. So this is perhaps the most important slide. So EPICERTIN is very different from conventional treatments, therapies for ulcerative colitis for the moderate to severe cases. So biologics and immunomodulators are available for moderate to severe cases of ulcerative colitis. EPICERTIN reduces inflammation similar to those drugs. However, unlike biologics and then immunomodulators, they essentially dampen immune system, the function of the immunity. Therefore, they run the risk of increasing, infections and malignancy. EPICERTIN doesn't have any immunosuppressive effect. Rather, it induces mucosal healing through recovering the epithelial barrier and then preventing the bacteria to get into the mucosal tissue, and thereby inducing mucosal healing. It's an epithelial repair agent. And that kind of activity cannot be attained by a conventional biologics or immunomodulators such as JAK and S1P inhibitors. And then also EPICERTIN can be administered topically, unlike other biologic monoclonal antibodies. And JAK and S1P inhibitors can be given orally, but it is absorbed into the systemic compartment, therefore have, again, the risk of infection and malignancy. However, epicertin does not get absorbed into the systemic compartment. It stays at the site of administration, therefore lower risk, lower systemic risk. Now, given the promising results in our preclinical models, we have been generating a lot of preclinical development data, including manufacturing development and analytical assays development, and also did additional analytical assay development and formulation development. And also we've collaborated with SRR Biosciences to perform non-GLP toxicity pharmacokinetic studies. They have been all published, and then we've generated a lot of data and encouraging results obtained and all published. So towards the first in human clinical study, our team include myself and a biopharmaceutical development specialist and a gastroenterologist and gastrointestinal pathologist, clinical health psychologist and mucosal immunologist, and then a regulatory consultant. We are trying to raise funding to perform first-in-human clinical trial at University of Louisville Hospital. And we've already submitted a pre-IND package to FDA back in 2020. We obtained written response from the agency. And good news is that they expressed no concern about the manufacturing of epicertin. They suggested to develop additional drug substance, drug product characterizations, non-clinical study design for non-rodent species. They suggested to use dogs, mini pigs, or non-human primates. And then clinical trial design, they suggested to go with a very conventional single ascending dose study in healthy volunteers. We've secured intellectual property for epicertin. We have 3 issued US patents, and also filed 2 applications, including 1 international. And I have co-founded a company called Grow Biomedicine recently, and then we received a phase 1 STTR grant from NIDDK. But currently, we are seeking to raise a total of approximately $8 million to complete preclinical development and perform a phase 1 first-in-human clinical study. We estimate that within 1 to 1.5 years, if we have the funds, we can submit IND. And then we are actively seeking an industry partner or sponsor or investors. So we got an STTR grant, but quite honestly, I don't have any other connections to go to raise the $8 million, and I'm hoping to have some partners, industry partners or investors through this event. Well, thank you very much for your attention. And this is my laboratory. Again, we are from University of Louisville, and appreciate your attentions. And if you have any questions, I'll be happy to answer.